Hello, we are Stability, the Tel Aviv University iGEM team. Today, we are going to talk about the problem that all of us synthetic biologists know exist but choose to ignore.
How dare you? Synthetic biologists don't ignore problems. We're perfect.
Greg, hear me out. So this problem is the evolutionary instability of genetically modified microorganisms.
Yep, that happens.
By genetically modifying a population of microorganisms for expression of a foreign gene, their fitness is reduced as they have extra metabolic load. This happens, for example, while manufacturing human insulin in yeast. Unavoidably, a mutation that reduces expression level of the target gene will arise. This mutated individual will now have a greater fitness and will eventually become the most frequent in the population, leading to the effective loss of the introduced gene.
But you can always use frozen stock.
You're right, Greg. But working from a frozen stocks allow only batch processes production, which decreases the maximum yield. It also greatly limits our ability to use genetically engineered microorganisms in an uncontrolled environment. Finding an elegant solution will help us to reduce manufacturing costs for biotechnology companies. More importantly, it will help synthetic biology revolutionize our life. One example will be using engineered microorganisms for in vivo drug manufacturing and delivery. In this case, genetic stability is of an immense importance, as the cultures are not replaceable and the safety of patients is of top priority. This is just a partial list of such microorganisms that are currently in clinical development. Live therapeutic agents are at the forefront of research, and our solution will be absolutely vital for their success. It is worth mentioning 2 papers published in Science about this last year. However, those solutions are highly specific and complicated to implement. Our solution is robust and is suited for a variety of purposes and organisms. Our solution is to transcriptionally interlock a desired target gene with an essential gene of that host organism, a gene whose activity is vital for the cell. This way, when a target gene is attached to the N-terminal of an essential gene, mutation that diminish the expression level of the target gene would be lethal for the cell, preventing such mutation from increasing in frequency in the population.
Wait, that means I can prolong the evolutionary half-life of any gene I want.
Exactly, Greg, and very easily. The way we link the genes together is dependent on your target gene intended usage and can vary from a simple linker to a sophisticated linker with a pseudoknot and a signal peptide, where a clean target gene is secreted from the cell. We set out to achieve 3 goals. Develop an AI software that customizes the best fitting essential gene and linker to your target gene based on bioinformatic model. Create a novel technique for large-scale measurement of genetic stability called GeneSeq. And finally, provide a preliminary proof of concept for our innovative solution. Throughout our project, we received feedback from our human practice engagement that further improved our design. Before we go into each step of our work, I will briefly describe a tool that helped us significantly in achieving these goals. Professor Maya Schuldiner created and graciously provided us with 2 libraries, one GFP-based and one RFP-based. These libraries contain 6,000 variants, one for each open reading frame in the yeast genome, with a distinct promoter per library. Each variant has a fluorescent protein fused to the N-terminal of a different gene. Using these libraries and our novel GeneSeq method, we prove the validity of our solution and gather empirical data for our model. And now, Karine will present the model.
Thank you, Matan. One main goal of our project is to develop an automated recommendation engine that, given an input target gene, would first match the best fitting essential gene to it, choose the best linker for attaching the genes, and optimize the combined construct for efficient translation and increased stability. For this purpose, we developed the following models. Our first model will help us rank the stability of different conjugated genes for a given target gene. We designed our model to predict the genomic stability obtained in our GeneSeq experiment, determined by the distribution of mutations within the target gene. Until we get these results, our model was trained on similar data, GFP fluorescence measurements of each variant in Professor Schuldiner's library. This prediction is based on many features we created using different data sources. To reduce the model's dimensionality, we selected a small set from these features based on a heuristic approach. Finally, we built a random forest regression model that recommends the most fluorescent genes, assuming their contribution to the target gene stability is the most significant. Consulting with experts and extensively reading the literature, we collected and calculated any feature that has a connection to genomic stability, such as protein-to-protein interaction, including PPI network analysis, codon usage bias, based on biophysical models of mRNA translation, and evolutionary conservation, based on molecular evolution models.
Sounds like a lot of work. How many features are we talking about?
It was a lot of work. We collected approximately 2,000 features, from which we selected 300 using a correlation-based metric. We evaluated the performance of our predictor when implemented on a test set. Our metrics of success are R-square, agreement on 20 leading candidates, and accuracy, which is defined by an error within 20 arbitrary units. As you can see, our results match the original ranking. Afterwards, our software finds the best linker candidates according to the construct purpose. For example, protein expression for intracellular processes requires using a fusion linker. This fusion demands consideration of both the folding of the linker itself and its effect on the protein's folding. We model the spatial disturbance that a linker caused to its attached genes by calculation of disorder profiles before and after the fusion using disorder prediction models.
That I understand, but what about the expression levels?
That's a good question, Greg. We meet the user's need for high expression levels by optimizing the construct for the entire mRNA translation process using 3 principles. First, regarding codon usage, the codons are selected for a high translation velocity based on empirical findings, the supply and demand of tRNA, or the usage frequency in the target organism. Moreover, at the 5' end of the open reading frame, there is a well-conserved signal for weak folding of mRNA. This signal promotes recognition of the start codon by the pre-initiation ribosomal complex. In addition, GC content is proved to affect the translation efficiency. The optimization framework we use is provided by DNA Chisel, and it allows us to utilize all 3 principles for efficient translation. The DNA Chisel algorithm detects contradictory constraints early and quickly reaches optimal solutions. Now, Itamar will present our software products.
Thank you, Karin. I will now introduce our second product and our main product's architecture. Our second product is the Evolution Failure Mode, or EFM Optimizer. It is also our 4th module and our submission for contribution. We based our product on the 2015 Texas iGEM team's EFM Calculator, a web tool in which you can insert a sequence and find mutational hotspots of the types simple sequence repeats and recombination-mediated deletions. You can carefully edit them and improve any sequence's genetic stability significantly. SSR sites are repeating short sequences. These could cause a polymerase slippage mistake. RMD sites, on the other hand, are long, identical sequences appearing in different locations within the genome. This could lead to a recombination error, where the intermittent genetic code is deleted. Recently, Professor Tuller's lab requested our aid in improving the genetic stability of a COVID-19 vaccine. The existing EFM calculator seemed like a good approach.
I don't know, running it on each sequence, fixing each site separately, sounds like a lot of work.
It is. That's why we created a whole new and improved platform. The original calculator could receive a single sequence at a time, consuming much time and effort. Now, with our user interface, it is possible to apply this analysis to many sequences at once. Not only that, in order to be more useful for mammalian and insectoid cells, we created a mechanism which finds the most likely methylation sites within a sequence. These are significant epigenetic inheritance hotspots, which increase DNA folding and reduce expression level. This improvement helped us advise the vaccine research further, as it was conducted on mammalian cells. By combining the EFM Optimizer and our optimization module, mutational hotspots found can easily be avoided in the optimization process we described earlier.
So you're telling me I can just take a bunch of sequences and your software will fix them for me?
Yes, Greg. Our EFM Optimizer scans all sequences within a directory for SSR, RMD, and even methylation sites. Then you will receive not only a ranked list of problematic sites, but also corresponding sequences optimized for stability and expression level. long, tedious manual corrections leading to non-optimal results are replaced by a click of a button.
Sounds great, but how does it fit with your main product?
Well, let's go over the whole process and see how everything clicks together. First, you insert your target gene into our software. This gene goes through our stability predictor and is matched with the best candidates for an essential gene. Then, according to your construct's purpose, our linker module provides the best linker candidates, according to the conjugated protein's folding disorder profile. At this point, the constructs are scanned for SSR, RMD, and methylation sites. Finally, the optimization module will present you with constructs optimized for stability and expression level, tailored to your needs. This concludes our modeling and software sections. However, this would not have been possible without empirical data to train from and verify our model. Itai will now present our first wet lab experiment.
Thank you, Itamar. We produced a new and innovative approach for measuring genomic stability. This method collects an enormous amount of data that is analyzed by our sophisticated model, as previously presented. This is our GeneSeq experiment.
GeneSeq?
Yes, Gene Stability Enhancing Quantifier. This experiment is the first test of our large-scale stability measurement technique called GeneSeq. It has two main goals. The first is large-scale testing of our hypothesis that the target essential gene linkage prolongs the half-life of the target gene. The second goal is to analyze the mutational footprints of each construct in order to eventually predict the preservation of the target gene. This is an overview of GeneSeq. The first step is creation of GeneSeq culture. For this first experiment, we used Professor Schuldiner's library to prepare two mixed cultures, one GFP culture and one RFP culture. Each culture consists of all the library's variants grown together. Afterwards, the mixed cultures were grown for approximately 300 generations using a state-of-the-art robotic platform called KeyBio. The KeyBio machine monitors fluorescence and automatically dilutes the culture with fresh media in order to maintain a constant optical density, or OD, of our choice. The OD and fluorescence are directly transmitted to the computer. After 300 generations, we extracted and prepared sequencing samples with our novel protocol. Then, we sent them for nanopore deep sequencing. The sequencing results could then be analyzed by our model.
Wait, is it even possible to sequence so many variants at once, both accurately and reliably?
Well, Greg, it is exactly where our GeneSeq protocol comes in handy. We extracted all DNA from the cultures. Then, we sliced the desired constructs using restriction enzymes, which were carefully selected by a restriction enzyme model that we developed for this objective. Our sliced constructs were treated with ligase enzyme to achieve self-ligation. We designed primers that would generate the PCR reaction only if the correct DNA sequence is ligated. Our sponsor, Cyclomics, a Netherlands-based startup company led by a former iGEM competitor, then made a rolling circle amplification of the samples and sequenced them in an Oxford Nanopore machine.
Wow, sounds complicated. Did we get any results?
Yes, Greg. Our first novel measurement technique for large-scale genomic stability was successful, and we are currently analyzing the results that were delayed due to COVID-19 related issues. Next, David will present our 10-gene experiment.
Thank you, Tai. I will now present our 10-gene experiment. This experiment provides a preliminary proof of concept. It validates that fusion to an essential gene likely prolongs the target gene's evolutionary half-life. In addition, it demonstrates that different essential genes provide varying stability levels. For this purpose, we compared the evolutionary half-life of various constructs against the half-life of the unattached target gene by measuring and comparing the relative loss of fluorescence. For this experiment, we selected 10 essential genes from Professor Schuldiner's library for our construct and applied an in-lab evolution experiment. We chose 10 genes by using clustering algorithms utilizing features previously described. We grew triplicates of each construct separately, where the unattached GFP was the negative control. We repeatedly grew them overnight, measuring fluorescence and OD for 180 generations.
So how did you choose your 10 genes?
Great question. We first filtered for essential genes, then for genes with no sensitivity to overexpression, and finally for those with a fluorescence above a certain threshold, resulting in only 600 possible genes. Finally, we implemented partitional clustering to find the 10 most diverse and representative genes for our experiment, based on the features previously discussed. And now, the results. As you can see in the graph, the x-axis represents the different essential genes used for our constructs. In green, you can see the negative control. The y-axis is the relative final fluorescence, normalized by the initial fluorescence. The negative control was second to lowest. This proves that linking the target gene to an essential gene prolongs its evolutionary half-life, and our solution works.
Wait, each construct behaved differently.
You're right, Greg. And that's why you need our software, to choose for you the best essential gene that gives the best results. Even more, the genes were selected for diversity and are not close to optimal for prolonging the GFP's evolutionary half-life. If optimized, results would have been far more significant.
That's amazing!
Thanks, Greg. Now, Adar will present to you our human practices.
Thank you, David. Our human practice efforts were directed at understanding our purpose and properly planning our vision. We created a workflow dividing our progress into 6 distinct directions. Industry. We had several meetings with some of the leading pharma and biotech companies in the world, helping us better understand the needs of the industry. We refined our product thanks to dozens of academic consultants. Entrepreneurship. We patented our solution. We impressed Lonza. They are currently helping us to build an initial business plan. We engaged the public with several lectures and workshops about synthetic biology and hosted the genomic stability workshop. We had many beneficial collaborations, including aiding many iGEMers in their research and hosting the second Israeli iGEM meetup. Collaborating with the other Israeli teams, we founded the Inclusivity Video Project in order to promote scientific aspiration in underrepresented communities in Israel. Last but not least, we ensured that each step of our research was done safely and responsibly. We used the ARIA framework listed on the RRI website. Using it, we refined the fundamental questions that need to be asked and answered in order to improve our project. Our efforts in integrated human practice helped us answer these questions. Who are our main consumers? All biotechnology companies and labs, but especially consulting companies like Lonza, DSM or Barents. How can we improve our solution for industry needs? By also addressing the issue of high expression levels in our solution and adapting it for secreted products. Most importantly, we understood why our work is so important and why should we keep working after the competition of iGEM.
I got a bit lost. Modeling, human practice, I don't know, I don't understand this. Could you go over it again?
No problem, Greg. Check it out. We met all criteria for iGEM Gold Medal. We devised an innovative model for predicting genetic stability using state-of-the-art bioinformatic tools and empirical data gathered from large-scale genomic experiments. We created 2 software tools. First, the genetic stabilizer that will revolutionize the field of synthetic biology and allow us as a community to dream bigger. And second, the EFM optimizer that has already proven to be useful to labs and other iGEM teams and will help many more in the future. We developed a novel technique for large-scale measurement of genetic stability. We provided a POC for our solution. We started a movement in the Israeli science community, aiming for a more inclusive tomorrow. And we succeeded in building a mutually beneficial partnership with a major biotechnology company that will help our vision come to life.
Heck yeah!
Thanks, Greg. We are also excited about being part of this amazing community. We want to thank all the people that helped us along the way, whether it was by consultation and generous donation or any other support. We encourage you to visit our website, read more about us, and learn how we can help you improve your product.